Hi, we're doing our presentation on neural circuits, and I'm Audrey.
I'm Francis.
I'm Aideen.
So essentially, our abstract is that because neurological diseases and psychiatric diseases are two of the most common types of diseases worldwide, and a lot of these diseases don't have cures or better treatment yet, we would like to study the complexity of the brain and its neurological circuits. So because of the brain's complexity, a lot of scientists have difficulty finding these cures for a lot of diseases such as Parkinson's and Alzheimer's. So in order to better the research done, we would like to use fluorescent microscopy to better picture these neurons and to fully map out the neural circuits, which are networks of interconnected neurons that impact behaviors. So with better research done on these neural circuits, we could potentially better treatment done on current diseases as well as be more prepared for future diseases that may arise.
The main question we will be addressing is how can we use fluorescence microscopy to better understand neural circuits for whole brain mapping? So the brain is one of the most complex and least understood organs, and 1 in 6 people have neurological diseases, and 1 in 4 people suffer from psychiatric diseases. Many of them currently classified or deemed as incurable, like Alzheimer's, Parkinson's, dementia, which can cause people to be bedridden, in wheelchairs, lose cognitive function or mobility. And through understanding more about the brain and its neurons, we hope to gain insight in not just one disease, but many diseases. And hopefully through this insight, create cures for the diseases. And this will help many people to take back their lives. And those affected will also be able to live with less pain and discomfort, hopefully. And for the more complex diseases, we'll hopefully be able to provide some help, and not necessarily a cure, but help them.
So currently there are many different ways to study the brain. So firstly, functional magnetic resonance imaging. It focuses on using strong magnetic fields and radio waves to create detailed images of the body, and it also researches the blood flow in the brain. So areas that have more blood flow than others are studied, they show that they're used more often than others. So transcranial magnetic stimulation. So what this is you have a magnetic coil on the scalp, and researchers will stimulate the neural circuits in order to alleviate psychiatric patients from their symptoms. So optogenetics, we were first introduced to this through simulating predatory switches in mice. So similarly, we could use light to simulate certain parts of the brain in order to have certain behaviors displayed. And nextly, positron emission tomography. This is also known as the PET scans. And tracking certain proteins with the radio tracers, we can see how it flows throughout the body and affects different processes. Deep brain stimulation has a device implanted in patients, and it sends electricity into abnormal parts of tissue, and it would reduce symptoms of damaged circuits. Both transcranial direct current stimulation and electroconvulsive therapy send electricity to the brain in order to help fix damaged neurons. And our last one, also known as DREADDS. So within neurons, certain proteins can be stimulated by different synthetic chemicals. So what researchers are doing are they're sending these chemicals in order to try to help fix these damaged neurons. So what we are proposing is that because certain parts of the brain are more understood than others, for instance, because Parkinson's and Alzheimer's are so common, there's more research done on those areas than others. So we would like to focus on the entire brain and try to map out the entire neural circuit system using fluorescent microscopy techniques. So we could tag certain brain proteins, such as using quantum dots, green fluorescent proteins, and etc., to help track where these neurons go and how they affect processes. So for instance, quantum dots by stimulating them with lights, the electrons can be excited in order to help fix damaged neurons. And similarly, green fluorescent proteins, when excited by lights, they can help neurons generate action potentials. And by understanding the neural circuit system, we would see how certain areas of the brain are connected to others and how certain processes affect others, and we might potentially find something that we might not have considered before. And moreover, we can also use thin slices of mice brain in order to visually see these neurons under these microscopes, because it has been shown that using mice to study human brains has been effective.
Okay, so upon doing this proposal, we might encounter some challenges, such as not being able to see the whole brain as a whole. And this would, not in complete detail, so this would be a challenge because we wouldn't be able to see how everything works in conjunction with each other. Also, confocal microscopes cannot achieve high-speed 2D imaging, and so this would cause the process to be very time-consuming, and results would come in long segments. Spatial resolution is any point source any point source of light that's smaller than the light diffraction, and when this occurs, there's a blur in the image, and so this can cause inaccurate images and not as detailed images, which would cause the results to be not as entirely detailed. And neuronal networks are dense 3D tissues, and because they're so packed and complex, it can be kind of difficult to study them and see what their structure is really like. Also, another challenge is observing a live specimen, it can go against some people's ethics, and so this would cause problems. Delivering light to the brain is also a problem by putting GFPs and quantum dots in, because they can react with the retina cells before going to the brain. So essentially, our purpose is to map out the entire neural circuit in order to have a better understanding of how neurons have an effect on how we behave and disorders and diseases people have. So by learning more about these different regions, we can pinpoint how different locations have an influence on how people react. And for future direction, we can develop better technology by carrying out this purpose in order to avoid the obstacles mentioned before. And this would not only help this research, but also future research or other current problems. And of course, we can break frontiers by discovering more about the brain.
So these are our contributions and our references, and we are open to questions. Yeah.
Do you have any specific diseases and associated specific areas that you think are understudied and that you would want to target first?
So dementia is a broad, it's a very complex disease, and it's also a very broad topic. So I think because it's so complex, there isn't as much research being done on it as potentially other diseases. So I guess that would be somewhere we could focus first. Okay, yeah?
So you are proposing to map out all the connections, right, in the brain. So it's a very valid proposal. It's actually going on right now as we speak in different places. So let's call it the connectome. So you are talking about fluorescent microscopy, but we know that it's really hard to image deep in the brain, right? So from everything we talked about and everything you guys have learned during these 5 days, how would you actually address that challenge of really imaging throughout an entire brain, that being like a smaller brain, like a mouse brain, or a bigger brain, like a human brain?
We might be able to use cryo-electron microscopy, because the electrons can pass through the sample and possibly see deeper into the sample than just fluorescent microscopy.
And also earlier this week, we kind of as mentioned by other groups, we looked at PET scans, which allows us to trace radio tracers throughout the body. So I feel like that would be very effective in studying how the brain flows and other areas like that.
So if you were to use electron microscopy, I think you will still have to somehow reduce the size of the brain, right? It's still a bit too big. So have you heard anything about how you could process your sample in order to do meaningful electron microscopy images on it?
So I think if we use electron microscopy, it would not, it couldn't be a live brain. It would have to be, it would have to be like, as we mentioned, slices of a mice brain. So I think in order to better study and prepare those neurons within those brains, as mentioned before, we have to freeze it somehow so that during the process of shooting electron beams, we won't damage any of those cells and potentially make an error within our research.
So you will have to slice the brain.
Yes.
Correct.
Yeah.
So can you think about another method we talked about where you would not necessarily need to slice your brain?
So this is the same question that I asked the last two groups, right? So we're going to answer it at this point probably if you guys don't have it, so we don't have to keep asking.
So
I can't think of it.
if you were to use fluorescence microscopy and you are facing that quite large object that is the brain, what have you seen, what have we talked about that can kind of help you out by modifying kind of the opacity of the tissue that you are working with? Remember those techniques to clarify tissues? Right? We can take a brain that is essentially opaque, right? We learned about that a little bit, and clear them out from everything that is lipid and that is causing scattering and opacity. So now you are dealing with a larger object, which is a brain, where all your neurons are colored in different colors, right? And now it's totally transparent, and you can imagine doing microscopy throughout the brain. Does it make sense?
Yes.
Yeah.
Yeah, so what I was asking two groups ago was multi-photon deep tissue imaging, which I know you guys heard about on Tuesday. And then this particular example is clarity, clarified tissues using light sheet microscopes, right, which you guys heard about, which is where you can scan large volumes very fast to get image maps, physical structural maps of the entire brain. So we won't have to ask it again, hopefully, but for those of you who think about it, well, the next groups, right, we learned a lot about a ton of stuff, right, and I know that was probably a small section of what you learned about on Tuesday, right, but the really advanced techniques, right, are really addressing some of these issues, making tissues basically transparent so that light can go through them, right, or using advanced multiple photon laser combinations to get deeper into opaque samples. And that was the type of stuff that we were sort of asking about. Like for, I think your group, when I was asking about the gut microbiome, right, you could image deep tissue imaging in a live rat using multi-photon fluorescence microscopy, right? And it's a live organism.
So since you are interested in the subject, you should know that there is a multi-million-dollar institute in Seattle that is called the Allen Institute, for which the sole